This last section is on genetic engineering. And genetic engineering is the manipulation of genetic material to have a desired outcome. We can actually transfer genes between different species. This is really easy to do with bacteria in the lab because, as we saw, bacteria can do some unique things with that horizontal gene transfer. They're also very easy to work with and they grow very quickly. So we can kind of take advantage of some of the cool things that bacteria do to help us to make some medicines, vaccines, and help us in different industries. So how does this actually work? So this is when we can actually fuse genes together. And we can kind of take advantage of those operons. If you remember talking about operons, that's going to be the way that bacteria can actually control their gene expression. So it had to do with that operator that's controlling the genes further down the line by way of that repressor protein. We could actually take genes and we could actually put them into that operon, and then we can actually control when those bacteria express those genes. So again, taking advantage of some of the cool things that bacteria have to our advantage. So if we wanted to get a product made, what we would have to do is that we would have to amplify the bacteria and the amount of product that they were actually making. So because bacteria grow very quickly and they're easy to grow in lab, we can actually grow a lot of bacteria and have that bacteria produce products so that we can get some proteins that we can use for vaccines, that we can use for medicines, antibiotics. We can also do this with detecting genetic defects as well as we are manipulating and doing genetic recombination in the lab. So we'll talk a little bit more about recombinant DNA technology. This is DNA that has information from two different species. So we're recombining different sources. We often call the offspring the transgenic or the recombinants. Again, we could take a gene of interest if, let's say, that we wanted to make a medicine, and if we inserted that gene within a bacteria, then that bacteria can actually incorporate that gene and then make that product. Oftentimes, the way that that gene is introduced is through some sort of vector. So it could either be through a bacterial phage as a vector or a plasmid as a vector. And then what we would do is that we would clone these cells, grow them up in large quantities, and then we could actually harvest that protein, refine it, and then we could use it for medicines or vaccines, etc. So how is this actually done? Well, we're going to have to cut out the gene that we are interested in that cell, and then we're going to have to incorporate it into the bacteria. And that's when we're going to use those useful things that bacteria do. Bacteria can open up and they can take in genes. And so that's a good way to introduce those genes so that that bacteria now has the codes to make that protein. So I think it's a little easier to describe from a picture. So this picture, and it is a very big picture, so I'm going to kind of move this around a little bit. So up at the top, if we have a gene of interest in a human cell, let's say that we want to manufacture some insulin to help people with diabetes mellitus. Insulin is a protein. So there's going to be a gene that will code for insulin, that protein. If we could isolate that gene and then input it into a bacteria, so now the bacteria has the genes to make that insulin, now that bacteria can make that protein and we can use it. So what we would do is we would isolate that gene. So we would use these molecular scissors, these endonucleases, to cut out that insulin from our human DNA. Then we could take a vector, the plasmid, and then what we would do is we would cut that plasmid and insert that insulin gene into the plasmid. If you notice with this picture right here of the plasmid, you'll notice that there is an antibiotic resistant gene. That's actually going to come in at the end as we talk about the growing of the bacteria and producing that particular protein of interest. So we'll talk about that at the end. So in this picture right here, you can see where we have the plasmid and we also have that gene of interest. Now we know that competent bacteria can open up and they can take in DNA. So in this case, we would combine this plasmid, bring it into the bacteria. And now we have our recombinant. So we have our bacteria that has taken in the plasmid, and that plasmid has our gene of interest, that insulin. Now, if we grow up this bacteria, it's going to be producing that protein by way of that gene expression. Now, remember, we are only interested in the bacteria that have that gene. So this is when that antibiotic resistant gene comes in. That antibiotic resistant gene was on that plasmid that contained our gene of interest, that insulin. So the bacteria, if we plate them up, if let's say that resistant gene was an ampicillin resistant gene, and we grew the bacteria in the presence of ampicillin, only the bacteria that are able to grow will actually have that plasmid with that insulin gene. We could isolate them, we could clone them, grow them up in large numbers, and then we could actually harvest our protein, the insulin, refine it, and then we could use it for drugs to help people. So what can we actually use this technology for, this recombinant DNA? So like I said, we can actually make important things to treat people. So insulin, if there are individuals that have clotting disorders, we might be able to make those clotting disorders. Antigens for vaccines. Maybe if people are fighting viruses, we can actually make interferon, which is a protein. We'll talk about interferon later on in the immunity chapters. I always say that interferon interferes with viral infections. So it kind of gives people that are fighting chronic viruses a fighting chance. It can help us to diagnose genetic defects. It can also be used for gene therapy. Gene therapy is where they can insert functional genes into cells to help cure diseases. Gene therapy is very successful, but is very targeted and also very expensive. And as you know, we can also use recombinant DNA for forensic science and solving crimes. Some other applications of recombinant DNA is in industry. We can use recombinant DNA to help us to ferment wine and beer. You tell me that, and you could have started with that because I'm sold. To help us to break down certain plant materials, help us with fuel manufacturing, can actually help us in a lot of different industrial applications. We can also use it in agricultural applications. So making crops with high yield seeds, maybe genetically engineering plants so that they are resistant to pests. Also controlling the pest population and insects. This is something that they actually have done with mosquitoes. Mosquitoes are often vectors for certain diseases. And in order to bring down the mosquito population, they actually genetically engineered mosquitoes so that when the genetically engineered mosquitoes mate with the native females in a population, the females actually go on to produce sterile offspring, and that actually drives down the mosquito population. This is actually what they did with many different diseases, one of them being Zika virus, which is transmitted by mosquitoes. And several years ago, there was the Summer Olympics in Rio, and this was the time that we were really concerned about Zika. And Zika virus was prevalent in that area, so they were really concerned about a large number of people from all over the world descending on this area and it possibly becoming a pandemic. Now, Zika is only carried in certain mosquitoes, not all mosquitoes. We don't have those particular mosquitoes in the Northeast. However, they did in the southern United States. And so what they did in many parts of the world is that they used these genetically engineered mosquitoes, and it actually helped to drive down the mosquito population. So around this time in Florida, they actually had some cases of Zika because they had that particular mosquito. So, of course, they were very concerned. There was public health concern. And so what they did was they sprayed with large amounts of pesticides, so much so that children couldn't go to school because of the conditions in the area. People couldn't go outside. And so they said, instead of spraying all of these pesticides that are harmful to everybody in the environment, why don't we introduce these genetically modified mosquitoes? Many of the people in the community were a little concerned because they're thinking, ooh, that's kind of like genetic stuff in a lab, kind of like Frankenstein in a lab. So a lot of people in the community were concerned. And so they ended up introducing those mosquitoes, and it drove down the mosquito population. It did not do anything to the people in the area. Of course, anytime that you are playing around with organisms in an environment, it can have some ecological issues, but it did drive down the mosquito population and drove down the incidence of Zika. We certainly have a lot of mosquitoes in this area. I can't even go out into my backyard because we have so many mosquitoes. Honestly, I would much rather have genetically modified mosquitoes introduced to my backyard than spraying a lot of pesticide, which then is getting into the ecosystem. It's causing harm with nature and the native organisms, as well as pets and people. So I mentioned that sometimes people hear recombinant DNA and they start thinking Frankenstein in a lab, and they start to get a little, kind of cringy about it. Well, recombinant DNA technology has been used for many, many years, and it is relatively safe. So no lab workers have become ill from known recombinant organisms. Usually the bacteria that they're using in lab, they don't usually infect humans. And because of those recombinant genes, it actually makes them less adaptable. And they found that using normal safety techniques like we would use in lab are fine for protecting individuals. So recombinant DNA has been around for a long time, and it's something that has done quite a lot of good for medicines, diagnosing diseases, as well as developing safe vaccines.